by using a next generation type of gene editing approach. So I'll tell you a bit about that. Let's see, next, that's it. So what we're all about is really to try to move gene editing beyond the two applications that are currently mostly being pursued, so liver applications, of course, but also ex vivo applications. I think if we all think about gene editing, these are really powerful modalities, right? CRISPR-Cas9, base editors, prime editors, but at this point in time, their commercial and clinical impact is relatively limited. And so our intention is to try to address this and hoping to deliver gene editors into the central nervous system. And we do this by combining a next generation editing technology that we have a proprietary access to through an exclusive license, together with our delivery platform, which is an exosome-based delivery technology, so a non-viral delivery system. We have a proprietary pipeline targeting primarily genetic drivers of neurological diseases. The first program is targeting ataxin-2, which is a gene that's implicated in ALS and spinocerebellar ataxia type 2, and also an MSH3 program for Huntington's disease. And these programs are rapidly advancing. We're coming up to candidate selection for the ataxin program this quarter, in fact. The MSH3 program is pretty close behind, and we're hoping to start CTA-enabling studies during 26 to be in the clinic in 27. And we have some really fantastic investors and partners, and I think we're all excited about the potential of the technology to really make a dent in some really, really difficult diseases. So I'll show you a bit of the data on the ataxin program primarily, but I'll talk a bit about the value proposition first. And really, exosomes, it's a natural nanoparticle. They're secreted by all cells, essentially. They're nano-sized vesicles, and that means that you tend to have a very good distribution within a given target organ. In our case, the focus is CNS, and we've certainly seen very good distribution in the CNS and into the CNS, but also to all the kind of key cell types in the brain, notably neurons, right, if you want to go after neurodegenerative diseases. And what's, I think a real differentiator also is that exosomes are, I think, a pretty proven modality from a safety standpoint. We have a lot of experience preclinically of taking exosomes into primates, but there's also been a multitude of clinical trials with exosomes, and normally those have been shown to be very, very safe and have good tolerability across the board, even upon repeat dosing, and that's also our experience. Another key differentiator for us is that we work on delivery of the RMPs, so say a Cas9 plus a guide or a base editor and a guide. And of course, what that means is that you get very rapid onset of action. The pharmacology is really quick, but importantly, especially in the CNS, you also have rapid clearance. So the exosomes and the editing machinery are not staying around in the brain for a long period of time, which of course, increases the risk of off-target activity. And if you compare that to AAV, of course, where you have persistent expression over a number of years, right? This is a very different safety proposition, we think, for patients. And of course, we're looking here at the natural nanoparticles, so we have an ability to genetically modify these exosomes to target them to different cell types. And so that's another, I think, really interesting attribute for certain indications. So as I say, the pipeline is very much focused on rare genetic diseases of the CNS. The lead program is targeting ataxin. We have an MSH3 program. Obviously, we're here today because we think there are some good opportunities to think about other CNS targets, but also to think about other extrahepatic applications of gene editing. And so happy to pick up with anyone during the conference or directly afterwards. The secret sauce, really, and the basis for our platform is essentially what you see on the left-hand side here, which is an exosome. It's a human natural nanoparticle. What we do is that we culture human cells in suspension, and we genetically modify them to secrete exosomes that contain a CRISPR-Cas9, CRISPR-Cas12, base editor, essentially the RMP, if you will, of a genome editor. And we do that through creating a fusion protein between an exosome protein and the drug cargo in question. And we have a cleavable linker in there. That means that we have a kind of a soluble RMP inside of the exosome, and that kind of shields the RMP, but also gives it really, really rapid uptake and delivery into target cells. So a very, very unique proposition, we think. And what's interesting about exosomes is that potency advantage. When we compare delivery using exosomes of an RNP to delivery using any other type of delivery vehicle, this is an in vitro context, we see essentially a 4 or 5 log superiority in terms of EC50 between an exosome and another type of delivery vehicle. And so the potency advantage, I think, is really key, and also plays into how we think about the application in vivo. And so when we run in vivo studies, this is just one example of a study where we were knocking out the TTR gene in the brain using CRISPR-Cas9 as a model system to try to understand partly potency, editing efficiency, but also biodistribution. We see in these rodents that we have very broad spread on both the contralateral and the ipsilateral side, but above all, we have very potent editing, globally speaking, across the brain. So we're not selecting cells here, we're looking at the brain in totality. And so we're getting up to perhaps 40% here in this particular study. So if you compare that to what you normally see with non-viral delivery approaches, it tends to be single-digit percentage and not particularly well distributed in the brain. And what we're seeing here is something that is completely different. And importantly, as I alluded to initially, we're also getting to all the key cell types. This is just a bit of a distillation of it, but in essence, we're getting to neurons, we're getting to glial cells, we're getting to all the cells of the central nervous system. If we compare that to, in this case, a VLP technology, which of course is another type of non-viral approach, you essentially have very little exposure to neurons, and of course that's absolutely critical for us as a differentiator in the case of neurodegenerative diseases. And what's interesting is that when we've done primate work and looked at the biodistribution in this study, we're looking at essentially a radio label of exosomes, tracking primates over 7 days after intrathecal and ICM administration. We're seeing overall a very broad distribution, which we think is pretty consistent with what we've seen in rodents to date. And so this is the starting point for our pipeline, and it's, as I say, very focused on neurodegenerative diseases. It's actually all trinucleotide repeat disorders, these 3 programs. In the context of SCA2 and sporadic ALS, we're looking at trying to edit the ataxin-2 gene, where you have a certain number of CAG repeats. In Huntington's, it's MSH3, and this is a DNA repair mechanism. We're a protein in those pathways, which is believed to be really key to driving somatic expansion of HTT, which I think is increasingly emerging as a really important target in Huntington's disease. In all cases, we see this as a one-and-done approach. The ability to go in and deliver an edit and then have a long-lasting effect, of course, is very, very attractive in these diseases. But we also have the ability to repeat dose, and of course, that's also critical if you need to top up the dose or try to improve the spread or get better exposure. That's the focus for the pipeline, and I'll show a bit of the data from the ataxin program in a second, but I'll stop on the platform nature of this to begin with, because I think it's interesting, right? When you have a platform, you really want to be able to iterate between different targets and between different types of modalities rapidly, and that's exactly what we're seeing here. We started our ataxin program just before Christmas, and we're coming up to candidate selection essentially in a few months' time. The MSH3 program has started, I think, in February or so, and we're already getting to a point where we have some really, really strong lead candidates that we're hoping to move forward. So, that's the beauty of a true platform, right? The ability to iterate rapidly and generate new products after products at a relatively marginal cost. So we're quite excited by that. And I'll show some of the ataxin-2 data. What we're looking at on this slide, and it's a bit technical, of course, is essentially on the microscopy pictures, we have untreated cells on the right-hand side, which are expressing ataxin-2, which is in red. And then on the left-hand side, or in blue, we see the nuclei of these cells. And when we treat these cells in the left-hand panel with our editing machinery, we see a complete ablation of ataxin-2 in this case, in a dose-dependent fashion, as you can see from the graph on the left. And these are at very, very manageable doses from a manufacturing standpoint. So we're really pleased with this, and I think this is kind of forming the basis for a lot of the candidate development work that we're doing at this point in time. And of course, importantly, when we do the same and just check that this is a specific ataxin-2 driven driven activity, and we use a control guide RNA targeting a housekeeping gene. We don't see any impact on ataxia, so showing us that we're on the right track here in terms of this program. And I'm not going to bore you with all the details on the development plan, but we have some important milestones ahead. Middle of this year, as I say, we're looking to select the lead candidate. By the end of Q3, we should have also selected the route of administration based on a primate biodistribution study that's currently being prepped. And then at the back end of 25, early 26, we're running maybe one, but maybe two, but possibly one, but possibly two in vivo proof of editing NHP studies, where we're hoping to see proof of editing in the primate brain. And of course, this is going to set us up really well for kicking off CTA and possibly also IND enabling studies in 2026. So I touched on the platform nature of this, and I think it's perhaps important to take a step back and zoom out. And what is exciting about the platform that we have is that we have been able to show that we can do exactly the same thing with different types of editing formats, whether we're talking about the original old school kind of meganucleases or TALENs, but also CRISPR-Cas9 and Cas12, base editors and prime editors, and of course, that gives a really good basis for working with others, for thinking about how you can create maximum impact. And the real beauty here is that the platform is very iterative. We're essentially plugging in a new RNP into the exosome, and off we go, so to speak. So a good starting point for a broader pipeline development effort. And I'll stop basically on manufacturing, and I think many people believe that exosome manufacturing is highly convoluted and a bit of a black box, but in fact it's becoming, I think, increasingly commoditized and now provided as a service line by several CDMOs. And in our case, the upstream process is very standard. It's a suspension HEK293 cell line that we culture internally. We have capabilities to do that up to 200 liters. It's very rare that we use that scale because it's not needed, but it's, you have that ability to scale it out if you need to. The downstream process, again, very bog standard. It's a combination of chromatography steps and filtration approaches that you would find in any CDMO working with biologics or viral vectors for that matter. But really the secret sauce, I think, in terms of manufacturing is analytics, and really to be able to get single particle analytics to understand, critical quality attributes, understanding how you can kind of inform both process and product development, but also ultimately how you can release material is really key. And I think we've invested a lot in building that analytical suite to enable us to move rapidly forward. So with that, I'm going to stop there and say that we're very excited by the pipeline that we have. We think that the initial data is looking very, very compelling in ataxia, but also in MSH3, and we hope that we can make a dent in these diseases. And certainly happy to talk to people about our programs, but also about other opportunities to collaborate and to partner. So thank you.